We will start. This time I will speak a few minutes in Spanish. I know that the Brazilians don't have any problem with this because you understand us. We don't understand the Brazilians or the Portuguese, so I will say a few words in Spanish, if it's okay for you. Quiero darle las bienvenidas a todos desde el Instituto Weizmann. Es un gusto enorme contar con la presencia de ustedes. Hoy nuestro invitado especial va a ser el profesor hSarel Flishman, que nos va a hablar sobre, es un investigador que nos va a hablar sobre coronavirus. Pero lo que me gustaría hacer es empezar contándoles algunas novedades del Instituto Weizmann y también, perdón, estoy cambiando acá el video, y también algunas novedades de lo que está pasando en Israel con respecto al coronavirus. Como ustedes saben, estamos en una serie de 3 eventos que los estamos haciendo todas las semanas, los días martes, terça-feira, y sobre temas diferentes. Esta vez el tema va a ser coronavirus, el tema del día, el tema del año, el tema de quizás de la década. Algunas novedades del Instituto Weizmann. Ustedes saben que hace 9 meses, 10 meses, en el Instituto Weizmann no había investigación referente a coronavirus. Hoy en día tenemos cerca de 65 grupos investigando coronavirus, en la búsqueda de las vacunas, en la búsqueda de los remedios, medicamentos o de soluciones epidemiológicas. Todos nosotros queremos soluciones inmediatas y eso no funciona tanto en la ciencia. Sin embargo, está pasando algo que no pasó nunca, que es un esfuerzo mundial increíble para conseguir soluciones. Es una de las pocas cosas buenas que están pasando y realmente en la ciencia hay una colaboración importantísima a nivel mundial. Muchos de ustedes participaron de un encuentro con Neil London, el doctor Neil London, hace unos meses, inclusive hemos hecho 2 para América Latina. Les quiero contar que el doctor London ha avanzado muchísimo en su investigación. Lo que él está intentando hacer es encontrar. El doctor Neil London ha avanzado muchísimo, hay muchos grupos que están avanzados, y les quiero decir que ha encontrado algunas moléculas que parecen ser muy efectivas con el coronavirus y están comenzando en test. En las próximas semanas, entre 4 y 6 semanas, van a comenzar los tests con animales y esperamos que muy pronto comiencen con seres humanos. Estamos hablando de un tratamiento, no estamos hablando de una vacuna. Sabemos que hay muchas vacunas avanzadas, pero también hay que encontrar tratamientos que puedan mejorar nuestra salud si nos enfermamos, porque el coronavirus es algo que nos va a acompañar y no va a desaparecer. Tampoco teniendo una vacuna no va a desaparecer muy rápidamente. Entonces ese es el primer tema. Tenemos buenas noticias muy esperanzadoras. El segundo tema que les quiero comentar es un poco lo que está pasando en Israel con el tema de coronavirus. Muchos se preguntan qué es lo que está pasando en Israel. No sé si saben, pero Israel ha sido considerado el país con más casos per cápita en el mundo de casos de coronavirus. Y seguramente todos se acuerdan o lembran que hace 6 meses Israel era uno de los países mejor posicionados en el mundo referente al coronavirus, con menos casos. Nosotros tuvimos el día de ayer más de 3.300 casos. Somos un país con 10 millones de personas. 3.300 casos es muchísimo. También hay que decir, juntamente con eso, que el día de ayer se hicieron 40.000 test en Israel, y de los 40.000 test salieron 3.300 casos positivos, ¿sí? Esa es la situación de Israel. Estamos con 467 casos graves hospitalizados, y el límite en Israel de los hospitales para entrar en una crisis total es de 700, 750 lugares. Está creciendo el número de casos graves, estamos camino a una situación muy problemática, que se sabe que si en un mes y medio no se toman medidas mucho más fuertes que las que se tomaron hasta ahora, vamos a llegar a una crisis de salud muy importante. Tenemos un problema en Israel, muchos se preguntan, bueno, ¿y qué pasa con el lockdown o por qué no se toman medidas más drásticas? Y la respuesta es muy simple. En todo el mundo se sabe lo que hay que hacer para bajar los números. Lo que está pasando es que hay un problema político en el cual principalmente los lugares más infectados son las ciudades ultraortodoxas y también los poblados árabes, y hay una cuestión de coalición y de manejo de gobernabilidad en Israel que dificulta la toma de decisiones. Esa es la situación que tenemos hoy en día. Hay manifestaciones en Israel, muchos dicen que hay problemas con la democracia y con muchos otros temas. Yo creo que justamente Israel es un país increíblemente democrático, con muchísima representación de diferentes poblaciones diferentes, y la razón por la que no se están pudiendo tomar decisiones es por el sistema político que no permite tomar decisiones, porque si no, si un primer ministro tomaría la decisión de que cierra todos los lugares problemáticos de ultraortodoxos, los ultraortodoxos están amenazando con salir de la coalición. Entonces probablemente lo que pase es que avancemos hacia un empeoramiento de la salud, de los lugares en los hospitales, y probablemente la presión finalmente no va a venir de los epidemiólogos, sino de los hospitales mismos, que cuando tengamos una presión muy, muy importante, se tengan que tomar decisiones drásticas. Esa es la situación hoy en día en Israel, un país en el cual no falta conocimiento para tomar decisiones, ¿sí? El país que es el Startup Nation, el país que tiene tantas personas con conocimiento y que investiga tanto, no está consiguiendo lidiar con la situación por una cuestión totalmente política, no por no saber lo que hacer. Esa es un poco la situación de Israel hoy en día. Tenemos esperanzas, claro, de recibir vacunas, pero entre todos entendemos que la vacuna por lo menos va a llevar 6 meses más hasta que la mayoría de nosotros la podamos recibir. Y también hay muchos científicos que recomiendan no tomar cualquier vacuna porque no se sabe exactamente hasta que sean aprobadas de una forma científica, ¿sí? Entonces hay que tener mucho cuidado también con salir corriendo a hacer ciertos movimientos que no tenemos la seguridad de que sean seguros. Les quiero comentar que nos está acompañando el día de hoy Carol Perelman de México. Carol es miembro de la Asociación Mexicana de Amigos del Instituto Weizmann. Carol estudió, vino acá al Weizmann hace muchos, quizás no tantos, porque no debería decir cuántos años, pero hace unos años vino a hacer el Summer Camp, el Bessie Lawrence del Instituto Weizmann. Luego volvió a México, se sumó a la Asociación de Amigos del Instituto Weizmann, creó, es fundadora junto con otra gente del Museo al Aire Libre de Ciencias que creó la Asociación de Amigos del Instituto Weizmann en base al museo que nosotros tenemos aquí en el Instituto Weizmann. Desarrolló muchísimos proyectos en México, creó también, entre otros proyectos, el grupo Vera Weizmann de Mujeres por la Ciencia en México, y vamos a tener el placer de que ella coordine la presentación primero del profesor y luego las preguntas y las respuestas. Carol, me gustaría abrir el micrófono, creo que lo tienes abierto, y antes de eso,
sí, ya lo tengo.
Antes de eso no me quiero olvidar de agradecerle a todos los directoras de las asociaciones por haber ayudado a organizar este evento, y mandar especial saludo a la doctora Silvia Gerson, nuestra presidenta en México, que hoy es el cumpleaños. Carol, el micrófono está contigo.
Dani, muchísimas gracias por la introducción tan generosa y gracias por invitarme hoy. I will speak in English so we can present Professor hSarel and start our meeting today. Thank you, Dani, for the introduction, the invitation, and I will gladly present Professor hSarel Flishman's exciting and very, very impressive professional career. Professor hSarel Flishman was born in Israel. He completed the Adi Lautman Interdisciplinary Program for Outstanding Students and the Life Sciences Research Track at Tel Aviv University in 2000. He then earned a Master's Summa Cum Laude in 2002 and a PhD with distinction in 2006, both at Tel Aviv University. He worked as a postdoc fellow at the University of Washington in Seattle from 2007 until joining the faculty at the Weizmann Institute in September 2011. Professor Flishman is working at the interface of molecular biology, biophysics, and biochemistry. He studies proteins, the working machines of living organisms. He and his research team developed computer algorithms that encode the current understanding of how proteins fold, bind, and interact, and then conduct experiments to verify the accuracy of their designed protein. Let me tell you that the Flishman Group was responsible for developing the first completely automated method for optimizing the activities of diverse proteins. Using these methods, they designed a protein that is an exciting candidate that may serve as the basis for an effective and very cheap vaccine against malaria. Such a vaccine could save countless of lives, especially in underdeveloped countries that suffer most from this disease. Currently, Professor Flishman is designing an antibody that will bind to the coronavirus and prevent infection. His lab had already studied SARS, which is like COVID-19 caused by a coronavirus. He was able to quickly apply his knowledge in developing computer models that is allowing him to design effective coronavirus antibodies, a work that could lead him to a vaccine. So after this presentation, please join me in listening to Professor Flishman's talk. But first, we will watch a short video about what Professor hSarel Flishman's COVID-19 research is about. And during the talk, please have your microphones off as you're muted right now, and send me all the Q&As, all the questions, the comments, everything through the Q&A button or the chat button at the bottom of your screen, so I could start looking at what everything we want to know, so we, at the end, ask Professor hSarel. So please, בבקשה, את הוידאו.
When this started happening, it was very clear to me that even though it's not a basic research question, this is the most important thing that I could do at this time. Over the past 8 years, one of the major focuses in our research group has been on learning how to design antibodies on the computer. And we've shown preliminary data that we can do this in atomic accuracy. We can actually design antibodies on the computer, then go to the lab and experimentally verify that they have the shape that we designed and actually have some of the functions that we designed. Antibodies are essentially our body's most important lines of defense against viruses. In our immune system, there are specialized cells called B cells, whose sole purpose is to develop antibodies to fight off infection and then to mass produce these antibodies. These cells become memory cells. And then the next time we face the same virus, we're essentially immune to that virus. This coronavirus is unique and new. Our bodies have never been exposed to this particular virus. And so when we face this new one, our antibodies are simply no good. In my lab, we had already started working on SARS, the previous pandemic. And so when this started, we were actually quite ready to switch over to coronavirus because the viruses are quite similar to one another. So we already knew what the vulnerabilities are of coronavirus. And so we're targeting these vulnerabilities with our tools. What we're doing on the computer is essentially docking all of the antibodies that we can lay our hands on against these vulnerabilities, and then we're refining the antibodies. Instead of relying on the immune system, we're refining the antibodies to bind more tightly. We know from SARS virus, again, that binding to these vulnerabilities can block infection. And in this process, we're producing millions and millions of different antibodies. And then we go to the lab and actually create, in a test tube, all of these millions of antibodies until we find a hit that's validated, and then we can provide it to our collaborators who will verify that the antibody is not just a good binder, but that it can actually block infection. We're trying to do whatever we can in the process of developing a new vaccine or even in treatment against this deadly virus. It's widely understood that this is a unique time. People are sharing both reagents, probes for experiments, their information, data. We got the molecular structure of the coronavirus spike protein before publication from our collaborator, just as a, it's not just goodwill. Can you do anything with it? He's also sending us material so that we can do the experiments in our lab. Once we're done with this, other people will pick our proteins up and verify that they are actually neutralizing. So this requires a massive interdisciplinary effort, and we're getting huge support from both within the institute and from outside. Everybody is trying to help, even though people are shutting down in some senses. Whenever you say this is a corona-related research, they say, okay, we'll prioritize this, and then they respond immediately. It's really a unique time and a very, I'd say, inspiring time for me as a scientist to see how the entire scientific community is coming together to solve what is probably the most important problem of our age, and where science can actually have such an immense impact on human society.
Thank you, Professor hSarel. And so please use your button at the bottom of the screen, the chat button, so you can send all the questions and comments. And bevakasha, Professor hSarel, if you want to, I can see you.
Oh, I'm here. So thank you very much, Carol, for this great presentation. And thank you also, Dani. My Spanish is very poor. My wife's grandmother was born in Maracaibo. And there are many family members coming in from Costa Rica and Venezuela and Argentina. So I can pick a little bit up. So I heard I give a really deep presentation of both the tragedy we're facing now biomedically, but also the political stalemate that's a great complication right now. So it is clearly a very, very challenging time. And even though our lab, as was mentioned in this brief video, even though our lab usually works on fundamental research questions relating to proteins, since this pandemic started, we've devoted a significant amount of effort to trying to address in any way we can some of these problems, and I'll provide an update relative to that brief video. So I'm sharing my screen with you now. Just a second. Can you see my screen? All right. Yes. The key question we're asking is, can we use our tools, can we develop new tools even, in order to address some of the problems in developing coronavirus countermeasures. These countermeasures can go towards developing better diagnostics, cheaper diagnostics, vaccines, stable and easy to produce vaccine, and also treatments such as antibodies to treat the disease in extreme cases. So I think probably many people have already seen these images of the coronavirus. The coronavirus gets its name from the fact that there are these protrusions coming out of its envelope, as you see here, these protrusions here. They're known as spikes. And the shape of this virus, people have imagined it to be sort of like a crown, looking up onto a crown. This is why it's called corona. And these spike regions, these protrusions, are really critical elements in how the virus invades our cells. These proteins first attach to cells that line our respiratory tract and then invade these cells, and this starts the infection process. So just a month after the corona pandemic started, the first atomic structure of this spike protein was already published, which was a record time for publishing an atomic structure of a viral protein. And this structure was key because it shows us some of the vulnerabilities, regions on the spike protein that we can target in order to block the infection process. So ever since we've seen this structure, we and other labs throughout the world have been thinking about ways in which we can target the spike protein in order to block infection. I'd like to start out by mentioning some of the challenges that anybody dealing with coronavirus and thinking about developing either diagnostics, vaccines, or treatments against the coronavirus, anybody thinking about these problems must really address. The first problem is that it's incredibly challenging to even mass produce this spike protein. It's a very complex molecule. I'll explain why in just a second. It's a very large molecule, very complex molecule, and turns out to be very expensive to mass produce, especially if you think about it. We don't need to mass produce it just for, let's say, 100 patients per year. We need to mass produce it for billions of patients. So this becomes a huge challenge, completely infeasible currently. The second problem is that if we try to optimize proteins, let's say to optimize the possibilities to express, to produce, to mass produce the spike protein, this is a very timely process. Because of the great complexity of the spike protein, of proteins in general, it's been known that in order to optimize proteins, it can take years. This is time we absolutely do not have. In this case of a global emergency, we need solutions actually now. We needed them a few months ago. So we need to move very, very quickly. So we can clearly see that the traditional ways, the current ways of thinking about how to optimize complex proteins are simply not enough currently. And this is where protein design, the field that my lab is working in, can come in. But first, I'd like to explain why is it so difficult to optimize proteins such as the spike protein. So first and foremost, proteins are really the most complex molecules we know. They comprise hundreds of amino acids. In the case of the spike protein, more than 1,000 amino acids, and that means tens of thousands of different atoms. They are highly structured. I showed the atomic structure of the spike protein last slide, and very, very sensitive to mutation. You can imagine that changing anything in that structure can cause the entire structure to unravel and break apart. We can also think about the numbers that are involved if we're trying to think about how to optimize some protein. So let's say we're working on a standard protein, nothing like the spike, which is huge, but let's say a standard human protein of just 300 amino acids. If you're thinking about a protein of just 300 amino acids, that means you're looking at the number of choices, the number of combinations, the number of solutions you need to evaluate is a number with 400 different digits. This is a huge, it's an astronomical number. It's actually beyond astronomical. It's more than the total number of particles in the universe. So obviously there's no way we can experimentally evaluate all of these options. It also turns out that we cannot even computationally evaluate all of these problems. So this seems like if you just think about these numbers, and again, the spike protein is much, much larger than 300. If you think about these numbers, this can be very discouraging. It means that essentially maybe there is no way that we can computationally optimize proteins. And this is essentially what attracted me to the field some 15 years ago, because this seemed like an unsolvable problem. So how do you solve an unsolvable problem? Obviously, you need a lot of computers. At the Weizmann Institute, we have several thousand different computers that our algorithms are using. When we need, we regularly make use of Amazon's cloud. But I think most important, and everybody will agree with me, is having a great team of people. And this is where I think it's just a privilege to work at the Weizmann Institute. We have really excellent students. I cannot stress this enough. None of what I'll show you in the following would have been possible without this excellent, committed, and intelligent team of people. We're also not alone. we're working within a global team of about 50 different labs around the world that are combining forces in order to develop protein design software. This is taken from a conference, an annual conference that we used to have every year near Seattle. Don't look for me in this picture. I think I'm behind the tree, so never mind. A few years ago, we were looking into this question of how to improve the stability of an existing protein, a naturally existing protein. So I mentioned earlier that the spike protein is unstable and therefore very difficult, very expensive to mass produce. So we were asking, can we, using computational methods, can we optimize the stability and improve the production yields of a natural protein? And Adi Goldenzweig, shown here, is a student who had worked on this particular problem and had shown remarkable results in the lab on a model system, a model protein, where she could really improve its stability and production yields. So then we asked, can we use this method in order to address a true biomedical need? And as Carol mentioned in her presentation, we worked on a malaria protein. So why malaria? I think to an audience from Latin America, I don't need to explain that malaria is still a very serious problem. It causes 500,000 deaths every year, mostly of young children. So it's really tragic. And even though there have been more than 60 years of research into developing a malaria vaccine, there's still no vaccine in clinical use against this terrible disease. Pardon me. A few years ago, this protein, which I show here as a cartoon, this protein is called RH5. It was discovered and characterized by a group at Oxford University and shown to be a really exciting candidate to serve as a vaccine, a potential vaccine against this terrible disease, because this protein, it doesn't function like the spike protein from coronavirus, but it has a similar idea. It's also essential for the process by which the malaria parasite invades our red blood cells, our blood cells, which are essential for the malaria parasite to grow in our bodies. And we know that by blocking, using antibodies in our body that are produced against this protein, these antibodies can block malaria infection. So if we could mass produce this protein and inject it to young children, pregnant women in Africa, this could go a long way in order to rid the world of this terrible disease. The problem is that RH5, this protein, is really notoriously unstable. It breaks down at temperatures as low as 40 degrees Celsius, and it's also very, very expensive to mass produce. And both of these are key problems for a vaccine for the developing world. Cost is obviously a problem, but also temperature sensitivity is a problem, because we must keep in mind that for this to reach the clinic, it will have to be mass produced and sent to countries in Central Africa where there's no electricity and therefore no refrigeration. So if the protein degrades at about 40 degrees Celsius, it won't be possible to use it clinically. So what we needed is to design a variant of this protein that is cheap to mass produce and is also stable in temperatures much above 40 degrees Celsius. So Adi designed 3 variants of this protein. We sent them to our collaborators at Oxford University, and they tested it. And within a few months, to our great delight, they returned with the following results. So first, it was very easy to mass produce this protein. It was produced in orders of magnitude greater amounts than the malaria protein RH5 itself. Obviously, we don't produce proteins in these chemical plants. We produce them in bacteria, as you see here. The design protein was also much more stable. It was more stable by about 15 degrees Celsius. So it was stable to about 55 degrees Celsius, which means that it can be safely mass produced and shipped even to the hottest regions on the planet. And third, and probably most importantly, is it immunologically active? Does it actually produce an immunological response as potent as does the natural RH5 protein? And the answer to that is yes. They mass-produced RH5, the natural RH5, and our design injected them to mice and showed that both of these proteins provoked the same immune response. The antibodies extracted from these mice could potently neutralize the malaria parasite in both cases. So this has recently been commercialized, and it's undergoing clinical trials, which is super exciting for us. And it also shows that protein design methodology, the methodology that our lab and other labs in the world are developing, this methodology can now address real world, not just model systems, real world vaccine development bottlenecks, not through an iterative cycle, but essentially in one shot. We were very excited by these results, and we wanted everybody essentially to be able to use the same algorithm. So Adi developed it into a really easy to use web server, which anybody around the world can just log into, provide their favorite protein. The Weizmann Institute cluster computes different variants over it for about a few hours, then sends back the results, and people can then do their experiments on their proteins. And I'm incredibly happy to see how broadly this has been adopted really all over the world, in Latin America, Europe, the US. And also this always warms my heart to see that this includes also countries in the Middle East, such as Saudi Arabia, Iran, and Pakistan, which is very, very, I find this to be very exciting. So this algorithm I showed you now addresses the question of protein stability and production. The complementary question, the next question that we asked, is whether we can use our approach also to design proteins with a greater level of activity. Could we improve the activity of an existing protein? And again, we wanted to test our ability to do that, not on a model system, not on a lab problem, a theoretical problem, but in a real world problem that may have an impact, a biomedical impact. So we turn to the problem of nerve agents. Nerve agents are these terrible chemical compounds that are synthetically made. They're not produced by nature. They're produced usually by terrorist organizations or by countries that behave like terrorist organizations to kill their opponents. And this has come into the news. It's literally in the news now. You may have heard about Alexei Navalny. This is a prominent opposition leader in Russia who was poisoned in Russia and has been treated by German doctors and is currently probably safe by one of these molecules. These are the chemical structures of some of these chemical agents. It's terrible that people are still using them, but it's a clear indication that we need a way to counteract these molecules. What you see here, never mind the chemical details, you can see though that these chemical compounds are quite different from one another. But remarkably, there is a naturally occurring protein called PTE that can break down these molecules. It's not great at what it does. It's actually quite slow. And that means that it cannot be used therapeutically. It's being used in labs all over the world to study various aspects of protein mechanisms, but it cannot be used therapeutically. So we asked the question, could we improve this protein and make it therapeutically useful? Can we accelerate or increase its rate of action against these terrible compounds? So we designed several dozen different variants, different mutants of this naturally occurring PT protein. And we went to the lab and tested whether these work better than the naturally occurring PT protein. And to our really great surprise and pleasant surprise, we found that quite a few of them improved their efficiency relative to the natural protein by, this is the fold improvement relative to the natural protein shown here in what's known as log scale. You can see that some of these designs improve the efficiency by 1,000 to 4,000 fold higher efficiency. And this is a remarkable level of activity. Actually, some of these, what I'm showing here, the different colors signify different molecules, VX, Russian VX, GD and GF, all these are nerve agents. And the efficiency relative to these nerve agents is shown here in these bars here, the pink, orange, and dark blue. And you can see that some of the designs improve the catalytic efficiency by about 4,000 fold. And this has reached, they've now reached the therapeutic level of activity, the level that's required for therapeutic efficiency. What's also quite exciting about these is that you can see that it's really the same enzymes, PT28, PT27.14, that are active against both Russian VX and GF. two different nerve agents, the same enzymes can break down both of them. And this is important because in any realistic scenario, if anybody is unfortunately attacked by nerve agents, we don't really know. The doctors won't know what nerve agents are being used until quite late in the process. So we need to have some sort of a cocktail of enzymes that can break down any nerve agent. And this seems like these particular designs can do this particular function. What's also very exciting about this is that it took us literally 2 months to reach these experimental results, compared to about a decade of work by our collaborators previously to obtain an enzyme that could be therapeutically relevant just for one of these compounds, VX. So it took us, using computational design 2 months relative to 10 years of work using traditional methods, and we've already reached the therapeutic level. And again, time is of the essence. If any of you has visited molecular biology lab, you've probably seen such messy images as you see here. Molecular biology and protein optimization are traditionally done in a very messy, iterative way, where you try many different options, you find what works, and then you carry on, do this one time after the other. And this is why these processes can take months and years. By contrast, what we've shown is now we have web servers in which anyone from around the world can log on, explain what they need to do to the web server, and then within a few hours, they get their results, and problems that took literally years and sometimes were simply not solvable can now be solved literally within a few months. And again, this is super important in the current state of affairs, where we're in the midst of a terrible pandemic. I'll explain what we're doing relative to the coronavirus pandemic. And we have several efforts. Really, a large amount of our work is now devoted to the coronavirus. Some of it is very long-term. Some of it is already seems to be bearing fruit, and it's very exciting. And I can't explain in detail all of it, but I'll just point out 3 different directions that we're working on. So first, again, this is the spike protein. There's a lot of effort worldwide to improve the production ability, the ability to mass produce the spike protein. This would be essential for the next generation of diagnostics against coronavirus. As we're trying to open up society and the economies, we need to know who's ill and who's been exposed, who's immune to the coronavirus, and having mass producing the spike protein will be critical for these efforts. Currently, the spike proteins, the naturally occurring spike protein is too unstable for mass production. We've designed a more stable variant of the spike protein, and the recent results we got from our Oxford collaborators were again very exciting. They saw an increase of about 10-fold in the production yield, and also an increase in stability. This is, again, very exciting. We think we can do better. And I hope that within a month or two, we'll have the final results on this. So this is one effort that may be relevant to diagnostics and vaccines. Another effort, I'd say worldwide.
hSarel, sorry. I just remember you that we want to allow Q&As. So you need to summarize, and we will allow the Q&As when you decide it, okay?
Yes. So I'll just cut to the point. Another effort that we're undertaking is to design our own antibodies, completely synthetic antibodies against these vulnerabilities. These are vulnerabilities that no human antibody is known to attack. And the reason we're doing this is because we know that coronavirus mutates. We also know that there are other variants of coronavirus out there, such as MERS. And we need a wide range of countermeasures against the coronavirus. This is so far not being successful. We have some indications. I'd say that this is a much longer term project for us. But again, it's very exciting, and we're very happy to work on this super important project. These are the people who are working on this. So I'll end by saying, thank my group, our collaborators, funding agencies, And maybe just when I stop and just before taking your questions, I'm actually much more interested in your questions than in talking. I'll just mention one last point, which is, I know that this is an incredibly stressful time. It's an incredibly stressful time for me as well, but I'm trying to be optimistic, because what I see is that science has made a huge advance, even over the past 6 months, of looking at this problem of coronavirus. There are already vaccines in phase 3 clinical trials. It seems like we'll have a vaccine in mass production already in the beginning, maybe towards the spring of next year. This is record time for any vaccine development. It used to be at least 8 years to develop a vaccine. Science has made a huge progress. There will probably also be antibody treatments already during the upcoming winter, which means that the death rate will drop significantly in hospitals throughout the world. So there's, even though this is a terrible, really a terrible pandemic, we should all take great care. I feel personally optimistic and also quite proud to be a scientist at this time. So with that, I'll close, and really I'm looking forward to your questions.
Thank you, Professor hSarel. Thank you for sharing all your knowledge. I want to stress the point that computational chemistry, which is what you're doing, is such an important field, because in the past, we needed to go to the lab and synthesize each and every one of the proteins that Professor hSarel is giving the computer the knowledge to do chemistry in a very cheap and fast way. And I want to start, there are a few questions, and I think that it's very important what you shared about malaria, because the knowledge from malaria helped you move to COVID. And I'm thinking of the Oxford group, which is doing the vaccine right now with AstraZeneca, had a similar approach, which they also have the malaria vaccine on the pipeline, and then they moved to COVID. So I want you to speak a little bit more about what one of the questions is about, which is, which are the best candidates for the vaccines, the COVID vaccines, and are these the same approach as yours?
So the vaccines that are currently in clinical trials are obviously based on, just to get to clinical trials takes months. So the vaccines that are currently in clinical trials are based on the naturally occurring COVID spike protein or components of the spike protein. These were done in an incredibly rushed way, just to get something into clinical trials in the hope that they would work. And I have, I think it's really our best hope for something quick. And I'm really hoping that these would work. The efforts that we've undertaken are meant to, in case that these vaccines show some problems. For instance, that they're not stable enough, that some subpopulations, some parts of the human population don't respond correctly to these vaccines, maybe even develop an undesirable immunological response. This sometimes happens with vaccines, and this is why it's so important to take care of doing, phase 3 clinical trials in a very, very careful way. There's been over the past month, there have been these publications about, or in the news, about the vaccine in Russia, which was rushed through phase 1, 2, and no phase 3. It makes sense to go through with these experiments very, very quickly. I think it doesn't make sense to completely drop phase 3, because we never know how a vaccine would be, what responses a vaccine would provoke in the general population. If we want to deploy this vaccine for billions of people, we need to know that there's no subpopulation, even at 1% ratio, that would adversely react to this vaccine. And currently, we don't know that about the Russian vaccine. I'm sure that the AstraZeneca Oxford vaccine, which is also being tested in Brazil, the Sinovac vaccine, I think it's also being tested in Brazil. Those are being done in a much more, I think, a safer, a more rational way.
There is another question here in the chat from Ariel Siegel. I will read it as it is. Is it correct that the vaccine will teach the immune system to produce the vaccine, and your research will produce the antibodies synthetically? Is that the main difference?
It's a great question. So there are there's more than two, but in general, you can think about two solutions that involve the immune system in order to address an infection. One solution is to develop a vaccine. That means you take the virus or the virus component that you're interested in. This can be the Rh5 protein or the spike protein, and you inject it into a human. The human immune system learns this protein, produces antibodies against it, and now this human becomes, it has the antibodies. Once it's infected with the real virus, it can deploy these antibodies and very quickly get rid of the infection. This is, I think, everybody thinks, this is the ultimate solution to a pandemic like coronavirus. This can be deployed at the level of the entire population. Is great. We also need countermeasures, solutions to the problem where some people do not respond to the immunological challenge. So they don't produce the antibodies. So let's say in some cases, we know that, for instance, in the flu, old people do not respond well to the flu vaccine. They don't produce potent antibodies against it. In those cases, when these become critical, we need another way of treating, and there are drugs against the flu exactly for these purposes. We need something like that also for coronavirus, and this is why people are also, including us, also developing antibodies against the coronavirus. So this question is really good. People are trying to do, and including us, to do both. Optimize antibodies on one hand, and optimize vaccine candidates on the other.
Yes, so one will create a natural immune response, and the other one are synthetic antibodies that can be used as treatment.
This is exactly, yeah, thank you very much.
Okay, okay. And then there is another question here, and I have my own question, which I will also throw at you. But Anna Flisser is saying, what Carol said is only the beginning, because in some moment the vaccine selected will have to be produced. How difficult will be this process?
Yeah, again, a really great question. I know one vaccine effort in a little bit more detail. This is the vaccine effort being done here in Israel in the Institute for Biological Research. And I think this thinking is replicated around the world in vaccine efforts. Their effort was meant from the beginning to go after a vaccine format that is known to be easy to mass produce and deploy in humans. So they looked at a vaccine that has recently been approved and actually shown huge efficacy in Ebola, in providing a vaccine for Ebola. And they've used exactly that format and adapted it for the coronavirus. So they took out the Ebola proteins, the relevant Ebola proteins, and inserted instead of them the spike protein to that format. And this is how they're going forward. They know that particular format they can mass produce very easily, quite cheaply. And this is why they're hoping that, they're now, I think they're planning to go into phase 1 clinical trials in October, after the holidays. This will take them time. Israel is a small country, but they're hoping that this will go smoothly through regulatory agencies and through clinical trials, and will be easy for them to mass produce for Israel and for Palestine. This is their goal, to be able to, in case it's difficult to get vaccines from AstraZeneca or for Sinovac, which will be first in line for sure, they'll be able to mass produce a vaccine for our region of the world.
Okay, another question. Does natural immunity to corona exist?
We don't know. I think there's speculation about this. I'll just mention this. I think the scientific community just simply doesn't know. There is some talk of what's known as cross immunity, yeah, with potentially other viruses. It's been observed that in Thailand, Laos, and Vietnam, the infection rate is quite a bit lower than in other countries. which raises some questions about maybe these populations have been naturally immunized by other coronaviruses. We simply don't know. There's probably a decade worth of research. Even after this pandemic is cleared, there's going to be a lot of research to answer these really important, I think, really fascinating questions about cross-immunity and other types of immunity. Currently, it's not known.
Yes. Okay, another question is, research has shown that T cells are important in the protective response against coronavirus. Is protein modification being done for more efficient T cell priming or activation?
This is really going deep into immunology. I'm not sure that I'm the right person with the right expertise to answer this question. In general, the response of the T cell arm of the immune system to a protein is also related to how different it is from human proteins. The spike protein is very different from any human protein. So I think there's no shortage of, what I know is that the immune system really observes, sees the coronavirus, it responds to it drastically, dramatically. A lot of, in convalescent patients who are recovering from corona, you see that a huge fraction of their antibodies is devoted to the coronavirus. It means that the T cell arm is really well activated. I'm not sure, from what I understand currently, they don't think that this is a problem. The T cell arm is actually activated in this case.
Yes, I think so too. How did the pressure to race against time looking for vaccines and treatments affect the work of the scientists?
This may be a personal question to me and
yes.
my group. So while the video that was taken and was shown at the start of this was during the lockdown of March, April. We were, at that point, everybody was, the Weizmann Institute was almost empty. It was very difficult to do any sort of work. I would say we felt that this was incredibly important. I felt very lucky, again, privileged, that it was very easy to ask management for help at that point. Whenever I said we're facing a problem, we need help in getting more computer power, we need help in resources, we need to open our lab. The key student on this project is French, he doesn't live on campus, there's no transportation, we need dorm facilities for him. Everything was done within hours. We had no problem in breaking through any sort of blockage at that point. They were very open to this. Again, I felt incredibly privileged at that point. And we all worked, days and nights to try to help as much as we can. Yeah, you do what you can.
Thank you for doing what you can. I want to ask you, there is a lot of media covering the llama microantibodies.
Yes.
So any relationship between those microantibodies, or what can you tell us about those?
Excellent question. The antibodies I showed at the end of my talk, these two things, these are nanobodies. These are llama-based antibodies. The reason we're so interested in these antibodies is that, okay, so just explain, antibodies from humans are very large proteins, which means that they have a problem interacting with small surfaces on viruses. And viruses often exploit that. They use this, the very large size of human antibodies to block them. So they put their most important parts, the most important surfaces in viruses are they're small and sort of sequestered such that our antibodies cannot reach. These are these two vulnerabilities I showed at the end of my talk. They're too small for human antibodies to reach into. So this is why we're trying to design smaller antibodies, llama or camel-based antibodies, to interact with these surfaces. So a method that is unique to our group is to develop, to be able to design nanobodies antibodies that come from llamas, but not use the antibodies coming from llamas, just use them as a basis for us to design new ones that can interact with, the coronavirus, let's say Ebola, other types of disease markers. Yeah, so this is, I think this is a very promising route for the future.
Thank you. The last question, and I will give the microphone to Danny, which is, what is your technical opinion regarding Russian vaccine? Well, you told us about that already, regarding a too-short phase 3. Okay, well, you addressed that already.
Yes.
A final comment you would like to share, and then Danny, thank you very much.
So yeah, final, just thank you all for your time, and this is, it's very important to have this interaction. I hope that next time we meet, we'll be with a vaccine, and that people will be able to travel safely and not feel so anxious about these things. So really, please try, take care. The social distancing and all that, it works. We should all take care until we have a vaccine. So thank you very much for your support.
hSarel, thank you very much for your talk. Thank you, Carol. I want to remember to everyone to tune in for the last talk next week on September 15 with Professor Meital Loren from the Department of Neurobiology. And now I will ask from Silvia, Dr. Silvia Gerson from Mexico, our president in Mexico, to close this session. Silvia, please. Do you hear us, Silvia? Please open, Silvia, please open your mic.
Thank you, Danny. Thank you very much to everyone. Good morning, Mexico. Good evening, Israel. I want to congratulate Professor hSarel Flishman for his very, very important work, and to thank him for sharing his knowledge with us, Latin American friends of the Weizmann Institute. Hearing you increases our pride and our gratitude to the Weizmann Institute for all the effort they do for mankind. Thank you, Danny Schmidt, CEO for Latin America. Thank you, Carol Perelman. Congratulations for your work this time and always. And we really hope the vaccine will be ready for spring. And we're wishing you lots of luck to your team, to yourself, and thank you for the time of everybody. This is inspiring us.
And now I will open we close this session, but the microphone is open if someone wants to stay. I will open the microphones anyway, but we finished this meeting. You can open your microphones if you want. Thank you all.
Thank you. Shana Tova.
Shana Tova. כל טוב לכולם.
תודה רבה.
תודה hSarel. Some of us will meet next week anyway, and we will have the opportunity to say Shana Tova again. But it's a great opportunity to wish every time Shana Tova and a good day.
And next year in the Weizmann in Israel.
Yes, but I remember you that this year we don't plan to do an international board with the presence of all of you, but we really will do an international board by this Zoom or an other platform, and we will have an amazing program in November, and we will see you anyway. It's what we can do until we will have the opportunity to take a plane and to travel again. Elias, you look good. I don't hear you.
Okay, it's pandemia look.
And I can say the same for Rick.
It's a different look, Danielle. It's a little bit different look. Shana Tova umetuka for everybody. It's great to see you, Danielle.
Thank you, Danny. Thank you to all the team at the Weizmann. And it really gives us hope to hear these talks and to be near the scientists and to listen to how they are approaching the details to uncover a solution. So thank you, Danny.
Thank you, Carol. Thank you.
Thank you.
And happy birthday, Silvia.
Thank you very much. Thank you for remembering.
Thank you. Happy birthday.
Silvia, you look younger every year. Maybe it's because Mauricio.
I'm sure. Thank you. It's really so important to have these talks and to be informed and to see the quality of your research. So thank you very much and keep with this work.
And to be in touch with everybody.
Yes, because this is a community. We really have a very nice community of friends. And, you know, we don't need to stay here to continue to speak, but we want to do it.
Yes, it is important. And my heart is filled with the hope and pride. So thank you very much.
Have a good day.
Thank you. Shana Tova.
Thank you.
Shana Tova.
Shalom. Shana Tova.
Shalom, Shana Tova.
Rolando, te acabo de... Ah, ahí cortó. Ok. Mándale saludos, Elías, a Rolando, por favor.
Claro que sí. Creo que ahí sigue todavía.
Ahí sigue, ahí sigue.
Sí, sí. Muy bien. Con mucho gusto.
Silvia.
Sí.
Chao.
Feliz cumpleaños.
Ay, muchas gracias, Vanessa.
Felicidades.
Gracias. Gracias a
Pase
todos.
bien.
Muchas gracias, claro que sí. Ya hicieron que mi día fuera memorable con esta plática tan interesante y tan importante. Así que muchas gracias. Un regalote.
Interesante especialmente para los químicos, ¿eh?
Sí, ¿cómo no?
Los químicos sí que lo entienden bien.
Muy, muy importante. Pero importante para la humanidad, creo, ¿no? En realidad es un placer escuchar este tipo de contribuciones. Gracias al Weizmann. Gracias, Dani.
Les dejo un abrazo a todos.
Gracias. Chao.
Chao.
Chao.